Welcome back, anatomy students. Today we're going to talk about the cell-mediated response. Most recently, we talked about the humoral response with B cells, and today we're going to do cell-mediated, which involves T cells. So the cell-mediated response involves T cells. That's the first difference between cell-mediated and the humoral response. The next big difference is that with the B cells, they can bind free-floating antigens, so they can directly bind them, and T cells cannot. They must be presented. So another cell has to have fractions of the antigens on their surface in order for the T cell to bind. So it has to be the antigen in combination with one of the cell proteins for it to recognize it. So typically what happens, a macrophage of some sort, a neutrophil perhaps, ingests an antigen and then processes it, so digests it, and then pieces of it will make their way to the surface in conjunction with one of the cell's proteins called a major histocompatibility complex. So that protein binds the antigen, takes it to the surface. That's what we see here with the HIV. So inside we have the HIV cell, and then fractions of that are going to make their way to the surface of the cell, and our T cells will recognize that on the surface. The antigen presentation, I think, is a difficult concept to recognize or to grasp, so I wanted to show you a couple graphics. Here you have a phagocyte of some sort, and it's eating up a pathogen, the red dot there, that's its antigen. So you can see here it's been digested, the red dot is making its way to the surface. So now that it's on the surface, it's said to be presented. And here you have a helper T cell that is binding that. Just like the B cells are very specific to the antigens they bind, helper T cells are specific, and each antigen has a unique receptor that binds it. This picture is a little bit more simple, but it involves the major histocompatibility protein, and so I like it for that purpose. So here you have some carbohydrate on the outside. It's being broken down inside of the cell, and then you can see pieces of it are binding the major histocompatibility protein, and then that protein follow it along. That protein will make its way to the surface. So the antigen is held in place by that MHC. And this is an MHC2, which is what helper T cells bind to. So you also have MHC1s, and that is what killer T cells bind. Okay, so helper T cells are considered the cell mediators, the mediators of the immune system. So they can trigger cytotoxic T cells to come and destroy cells, or they can trigger B cells to make antibodies. And what they're really doing is identifying cells that belong to you and cells that don't belong to you. So they have different receptors on their surface. So these that kind of look like a hashtag and these that kind of look like V's. So the hashtag would recognize any of my own cells. So my receptors on my cells, my antigens, bind and match this receptor. Any binding antigen that is different than mine has a different shape. So this is kind of like, you remember we talked about the A antigens and the B antigens on your red blood cells. So this would, I'm an A positive blood. So this could be an A positive antigen. It would just be an A antigen. And this might be a B antigen, and my body recognizes that as not belonging. But it might also be an antigen from a virus or from salmonella or something like that. So the T cell goes around and it bumps into your cells, and it decides whether or not that cell belongs to you, or if that cell is an invader, is foreign. So you have 5 different T cells, and we're going to be pretty simplistic with this. You have cytotoxic killer T cells, and these are often down here. You might notice it's denoted with T sub C. So you could write it that way. They're also called CD8 cells. You can see that there. So the cytotoxic killer T cell works very much like a natural killer T cell. So it injects perforin into the cell, and this is a foreign cell, and these are its antigens on the surface. And then the purple dots are the perforin. So the cytotoxic killer T cell injects perforin into that cell, which causes pores and causes fluid to rush in order to lyse it. At the same time, the killer T cell is releasing an enzyme called a granzyme. The granzyme goes inside those pores as well and causes apoptosis. So that is how the killer T cell works. So the killer T cell is in charge of the destruction of foreign invaded cells, which might be virus-infected cells, cancer cells, or a transplant cell. And like your B cells, they're found in your lymph nodes, spleen, and tonsils, and they'll receive like a chemical signal that will trigger them, and then they'll follow that to the site of inflammation, and that's where they'll do their dirty work. They'll inject their perforin and get rid of cells that don't belong. The helper T cell, the most important maybe cell, is in charge of connecting and identifying what type of antigen is present. So here is some antigen presenting cell, a phagocyte of some sort, the helper T cell here, and then it will decide if this is an intracellular threat or an extracellular threat, what type of a response needs to happen. So if it's an intracellular threat, we're going to signal the cytotoxic killer T cells to come to the area. But if it's a humoral, if it's a extracellular threat, it's going to signal B cells to come to the area, and then B cells will proliferate and make their antibodies. So this is the cell that is targeted by HIV. HIV targets the helper T cell, destroys that cell, and so you can see why our bodies are susceptible to infections that we typically don't succumb to, because our body is normally fighting off these infections. So they're opportunistic infections. They take advantage of the fact that your immune system is weakened. So the helper T cell, because it's not there, is not able to signal the rest of your body to respond. And so those infections take advantage of it. So this is just another graphic. Here is an antigen being presented. A killer T cell might bind directly. So the killer T cell binds MHC1s. So on your infected cells, they might be presenting their own antigens. Helper T cells bind MHC2s. So those are on our phagocytes. And so then the helper T cell will either send out one cyclin type chemical that signals the B cells to proliferate and make antibodies, or it sends out a different chemical that signals the T cells to proliferate, and then they go and do their thing. They destroy your cells. So the third one, once we've fought the good fight and we've won, the suppressor T cells kick in. So to conserve energy and resources, we don't need to keep fighting an infection that's no longer there. So the suppressor T cells will release chemicals that suppress or stop B and T cells from proliferating, and we will no longer respond. The last two helper T cells are the delayed hypersensitivity T cells and the memory T cells. So delayed hypersensitivity, delayed, it happens later, right? So maybe 12 hours before these cells kick in. And hypersensitive, hyper is above or in excess, right? So you're being overly sensitive, you're overreacting. So T cells are responding to things that shouldn't be harmful to you. So this is your allergies, so playing a role in allergies and prolonged inflammation. So like pollen truly should not be harmful to us, but our body overreacts sometimes and thinks that a pollen grain is a bacterial infection, and then it mounts a response. And so quite often that has to do with the inflammatory response. We have swelling, we have itchiness from it pressing on nerves, there's heat, and poofiness just from the leaky vessels. So delayed hypersensitivity has to do with your allergies. And then memory T cells, just like you have memory B cells, when the helper T cells proliferate, they make some that are going to be used now, some that will last for years and be used later. Same thing with your cytotoxic killer T cells. Some will be used now, others will become memory cells to be used later. So that aids in our infections for the future. So that's all I have for you on the cell-mediated response. Hopefully that was short and sweet. The T cells, you can name the 5, right? So you have helper T cells, which signal cytotoxic killer T cells, and then they will be suppressed, and we will form memory cells, and hopefully you're not bothered by the delayed hypersensitivity cells, because allergies are not any fun, right? So anyways, thanks for listening. Bye.